Hello, this is Paula Tible and this is my PowerPoint on how to save the mangrove finch of Galapagos Islands. Camarhynchus heliobates, or the mangrove finch, is one of the 13 species of finches described by Charles Darwin when he studied them in the Galapagos Islands. The Galapagos Islands biodiversity has most of the original species that existed there prior to human colonization. But because of human colonization, the introduction of foreign invasive species has occurred, and thus many native species, including the mangrove finch, has become endangered. The mangrove finch is the most endangered of all of Darwin's finches. It's considered critically endangered. The mangrove finch used to inhabit the islands of Isabella and Fernandina, but now it's only found on Isabella in two small locations on the northwest coast and an extremely small location in the southeast coast. The estimated population is about 100 birds. Population decrease due to predation of introduced species has occurred, and those species are the parasitic fly Philonurus downsii, paper wasps, and fire ants, and also animals such as cats, black rats, and smooth-billed ani. Seen here are the two introduced species that will be important to the study of how to save the mangrove finch from extinction. Shown here is a black rat which consume mangrove finch eggs while they are incubating. Also shown here is the Philonurus downsii, whose larvae prey on finch younglings, as shown by the picture. The larvae suck the young's blood, and this is very detrimental because it causes lower weight, tissue damage, and anemia, which in the long run can lead to death and bill distortions. Materials and methods. 4 locations of Isabella Island will be studied from 2006 to 2009. Caleta Black, Selvita, Playa Tortuga Negra, and Bahia Cartago. Rat control will occur in some locations in November 2007 at Caleta Black and Playa Tortuga Negra. The control group is at Selvita, in the middle of the two, where rats are trapped and then recorded and released to represent the population where no active efforts were done to control the rat population. False nests with false eggs will be used to monitor rat predation. The false eggs were made of modeling clay, and markings on the clay will determine if the egg was attempted to be eaten by birds or rats. Actual finch nests will be monitored for reproductive success. Probability of species survival will then be predicted using 4 different scenarios. Here is a map of Isabella Island as to show where the 4 locations are on Isabella Island. As you can see, there is Caleta Black, Selvita, Playa Tortuga Negra, and then on the east side of the island there will be Bahia Cartago. These are the 4 possible scenarios for the future of the mangrove finch. No intervention was done, low intensity rat control, high intensity rat control, high-intensity rat control, and control of Philonurus downsii. All 4 scenarios were then run over 100 years and with 500 repetitions, so a mean probability can be created. Rat control results were that Selvita had 87% predation in November 2007, and since no rat control was conducted, in January 2008 a high percentage still existed, which was 73% predation. Playa Tortuga Negra had 77% predation in November 2007, and after rat control was done, in January 2008 it had a low percentage of 28% predation. All of the predated eggs in Selvita had rat tooth marks, while 50% of predated eggs in Playa Tortuga Negra had rat tooth marks, so the other 50% could be assumed to be done by other predators. Finch population in 100 years. This was modeled by using Vortex version 9.92. No intervention was done, If no intervention was done in 100 years, mangrove finches will not exist. If low-intensity rat control is done, the population will stabilize at Playa Tortuga Negra in 100 years, but other locations there will be no more mangrove finches. If high-intensity rat control was conducted, the population will stabilize at Playa Tortuga Negra and Caleta Black, but at Bahia Cartago there's only a 50% chance of mangrove finches existing there. If high-intensity rat control and also the control of Philonurus downsea occurred, the population will stabilize at all three locations. Conclusion. Poisoning the rats to control their population was successful in reducing rat population and their effect on mangrove finch nests. In the prediction of finch populations in 100 years, some factors were overlooked. Because of tropical storms on the breeding practices of the finches can occur, that factor was ignored. Also, the other factor that was ignored was Philonurus downsea infestation can occur due to more nests lasting to the nestling stage due to the lower population of black rats. There is also the factor of reduced genetic variation in mangrove population. This can also be a factor, as such a decrease in diversity can result in a loss of evolutionary potential for the mangrove finches, for it makes them less likely to adapt to environmental changes. Here are my references, and thank you for watching my presentation on how to save the mangrove finch.